Hi everyone, my name is Paige and thank you so much for joining me for a lesson on the cytoskeleton. So the cytoskeleton is a cytosolic protein filament network, and it has a variety of functions, namely transport, stability, movement, and cell division. And there are three different kind of flavors of filament within the cytoskeleton. So we'll start with the smallest filament, and that is actin. These filaments are about 7 nanometers, and they're the central component of microvilli and stereocilia. However, you'll see them in a variety of different cell types. And the way that the actin polymer is built is from G-actin monomers, they're called. And with the energy from ATP, they will form polymers called F-actin. And two of these polymers will spiral together to form a double helix that is sort of the final actin structure. So let's zoom into our image over here to see what that looks like. So let's say that this is the cell, and here's the nucleus. Along the periphery out here, you can see depicted actin. And here's the depiction of what the filament itself looks like, and you can see that it is a double helix made up of two F-actin polymers, which are made up of G-actin monomers that polymerize using the energy of ATP. And if this looks a little familiar to you, take a look at the diagram up here. This is a picture of a contractile unit in the muscle. So here you can see actin in pink here, and then another motor protein called myosin. And myosin here forms cross bridges with actin to cause contraction. So what'll happen is you can see all these tiny little heads here on the myosin that are going to interact with the actin, and it's going to cause this contractile unit to shorten. So that myosin motor protein, when associated with actin, is really important for the action of muscle contraction. Actin is also involved in migration and cytokinesis. So you can see the proximity of the actin to the periphery here, and finger-like projections can extend off of the cell in a single direction to help the cell crawl to where it needs to go. And since we did just talk about the contractile power of actin, we can see how it might be used in cytokinesis. So here, let's say we have a cell that's splitting into two cells. Actin can form a contractile ring right in the middle to help squeeze those two apart and help with the division process. Let's move on to the next biggest filament type, which are called intermediate filaments. They're about 10 nanometers, and their primary function is stability. So they don't associate with motor proteins like the other filament types do. Instead, they're mostly just there to hold down the fort. And as much as I would love to tell you about the structure of intermediate filaments, we would be here all day. That's because these intermediate filament structures vary from cell type to cell type. And now, while I did mention that intermediate filaments don't associate with motor proteins, because they are there for stability, they do have accessory proteins called plectin, which help to cross-link them together. So if we zoom into our image here, we can see all of the purple intermediate filaments, and they would be cross-linked, like so, by plectin. And now there are a lot of different types of intermediate filaments, as I alluded to, based on the cell that they're in. And these are names that you're going to want to be familiar with. So cytokeratin is found in epithelial cells, vimentin is found in fibroblasts, desmin is found in muscle, glial fibrillary acidic protein, or GFAP, is found in glial cells, and lamins are found in the nuclei of cells. And the reason you want to know all of these names is because the immunohistochemical tumor markers for each of these can be used to detect the origin of a bunch of different neoplasms. For example, GFAP positivity is really classic in board questions for glioblastomas, as well as sometimes astrocytomas. And that same concept applies to a lot of these. So cytokeratin can be seen in a lot of skin cancers. Vimentin can be seen in sarcomas, meningioma, and mesothelioma, basically mesenchymal cells. Desmin positivity would be a marker for rhabdomyosarcoma, etc. So if you understand what these markers represent, you can determine the origin of these tumors. So let's move on to microtubules, which are by far the largest of the cytoskeleton filaments. They're about 25 nanometers in diameter, and these make up the core of cilia and flagella, as well as playing an integral role in the mitotic spindle. And similarly to actin, microtubules are going to associate with motor proteins. However, in the case of microtubules, it's two motor proteins called kinesin and dynein. So let's zoom back in and look at those. So in order to understand how these microtubule motor proteins work, it's important to understand that microtubules are polarized. So as you can see here, all of these green microtubules come together to a central point here right outside the nucleus, called the microtubule organizing centers. And the end of the microtubule that is anchored near the nucleus is called the negative end of the microtubule. And then by default, the other end of the microtubule that's out by the periphery is the positive end. So negative near the nucleus, it's the N sound. Positive towards the periphery is the P sound. That's how you can remember it. So here we have a zoomed in version of a microtubule where you can see the two motor proteins, kinesin and dynein, walking along carrying out their motor activity. So kinesin is responsible for anterograde transport. So it'll be moving from the nucleus, the negative end, towards the positive periphery. On the other hand, dynein is responsible for retrograde transport. So it'll be going from the periphery back towards the nucleus. And now several different pathogens can hijack dynein to take advantage of that retrograde transport towards the nucleus. So polio, for example, will latch onto dynein and use it to take itself towards the central nervous system. And other pathogens that do this include Clostridium tetani, herpes simplex, and rabies. And all of those, again, are using it for its capacity for retrograde transport back towards the nucleus of a cell, compared to kinesin, which was used for anterograde transport towards the periphery. Now, there's a class of drugs called microtubule inhibitors. This includes mebendazole, griseofulvin, colchicine, vinca alkaloids, and taxanes. So there's quite a few of them, and they run the gamut between anti-helminthic, antifungal, anti-gout, anti-cancer. And in order to understand how these work, it's important to understand the structure of a microtubule in a little more granular detail. Now, microtubules are made up of these dimers called beta-tubulin, and microtubules are famous for getting longer and shorter by polymerizing up or depolymerizing down. So you can see the process of polymerization here, where each of these dimers is attaching on to make the microtubule longer, and the process of depolymerization, where the microtubule will rapidly just sort of fall apart and get significantly shorter. And microtubules are constantly polymerizing and depolymerizing. It's how they do their job. Now, most of these drugs, the first four here, are going to work by inhibiting microtubule polymerization. However, there is a very high yield exception to this, and that is taxanes. Taxanes, instead of inhibiting polymerization, stopping the microtubule from being able to grow, instead, taxanes are going to promote the stability of the microtubule. In fact, they're going to make it so stable that it can't depolymerize. Instead, it's just going to be stuck where it is, and it's also not going to be able to do its job. So while vinca alkaloids and taxanes are both microtubule inhibitors used against cancer, they have two very different mechanisms of action within the class of microtubule inhibitor. So that's just a high yield point to be aware of. However, in both of these cases, inhibiting the microtubules is going to cause failure of the mitotic spindle. So the lengthening and shortening of these microtubules is really important in cell division. And so inhibiting this is ultimately what makes these drugs effective. Now, inhibition of microtubules as a general rule tends to cause peripheral neuropathy, specifically in the cases of vincristine, which is a type of vinca alkaloid, and the taxanes. Now, the reason I said vincristine specifically is because another vinca alkaloid called vinblastine does not cause peripheral neuropathy, and that's another little pearl they like to throw at you sometimes, so just be mindful of that fact. In addition to drugs that inhibit microtubule polymerization, there are also disorders that inhibit microtubule polymerization. For example, one called Chidiac-Higashi syndrome, which is an autosomal recessive disorder in the lysosomal trafficking gene, or the LYS-T gene. So let's say that we have a cell here, and this cell has just phagocytosed a bunch of particles. So this right here is the phagosome. And after phagocytosis is complete, microtubules are generally going to be what facilitate the fusion of the phagosome to the lysosome. And if you're interested in all the different presentations of this disorder, there is a separate video where we go into this condition at length. However, an interesting presentation of this is that the patient will have peripheral neuropathy, just like with some of those microtubule inhibitors. So in conclusion then, there are three different types of filament that make up the cytoskeleton. Actin is the smallest with a 7 nanometer diameter, used in muscle contraction as well as cytokinesis and migration, and it's structured in a double helix that associates with myosin. Intermediate filaments are about 10 nanometers in diameter. They're used in stability. They have widely varying structures and will cross-link with the protein plectin. And microtubules are the largest, about 25 nanometers in diameter, used mostly in movement and cell division. They have a cylindrical structure that's constantly polymerizing and depolymerizing, and they associate associate with the proteins kinesin for anterograde transport and dynein for retrograde transport. So that's all I got for you. Good job, good luck, and keep practicing.